Welcome dear students in MSc Second Organic Chemistry class. In today's lecture, we are going to learn the NMR spectrum of decalin system. In previous lecture, we know that we learned the ring clipping or ring inversion concept. This NMR spectrum of decalin system is correlated to the ring inversion or ring clipping concept. How? Let us consider this trans-decalin system. We know that this trans-decalin system is a rigid structure and it does not convert it into the one form into the another form. We can say the ring clipping or ring inversion is not possible in case of a trans-decalin system. This is a ring, this is B ring. This A ring contains, suppose we consider the carbon number 1. This carbon number 1 have axial hydrogen, equatorial hydrogen. Carbon number 2 have axial hydrogen, equatorial hydrogen. Carbon number 3 also have this equatorial hydrogen, axial hydrogen. Carbon number 4 also have equatorial hydrogen, axial hydrogen. Carbon number 5 also have equatorial hydrogen, axial hydrogen. Carbon number 6 also have axial hydrogen, equatorial. Similarly, carbon number 7, 8 also contains one of the hydrogen as axial and another as equatorial. So we can say overall in this molecule there are 10 hydrogens are present in axial position and 8 hydrogens are present in equatorial position. So we know that as the structure, this is the trans-decalin system, as we know that, and we know that the trans-decalin system does not undergo ring clipping, means trans-decalin system never changes one conformation to the another conformation. Means supposed to be this is a 9 carbon, this is 10, then 9C3 conformation is never converted into 3C9 conformation, because the joining of carbon number 5 and 8, this carbon number 5 and 8 after ring clipping is not possible. This concept is known in previous lecture. So 10 hydrogens are present axial position, 8 hydrogens are equatorial position. But ring clipping is not possible. Therefore, this equatorial hydrogen and axial hydrogen shows coupling with respect to each other. And because of this coupling, we observe that there are some broad lines. Some means these such type of broad lines are observed in NMR spectrum. The hydrogen will shows a coupling with the equatorial hydrogen, and therefore it shows broad spectrum or broad spectral lines in NMR spectrum, because the rotation is restricted. But when we compare this molecule or when we compare this NMR spectrum with the NMR spectrum of the cis-decalin system, when we compare this NMR spectrum with the NMR spectrum of the cis-decalin system, then we clearly observe that the NMR spectrum is different. We know that in cis-decalin system, a ring inversion is easily carried out. Suppose I carried out the ring inversion of A ring. We know that this is carbon number 9, 10, 1, 2, 3, 4, 5, 6, 7, 8, like this. So this conformation for a ring, this conformation is 9C31. After ring flipping, it is converted into the, suppose I carried out the ring flipping of a ring, then 9C3 conformation is converted into 3C1, 3C9, means this is carbon number 3, this is 2, 1, 10 and this is 9. Sorry, this is 9 and okay. So this is carbon number 10, this is 9. Okay. So after ring flipping, we observe that there is a formation of such type of structure. Means ring flipping is easy. We know that this ring flipping in cis-decalin system is easily carried out. 9C3 conformation is easily converted to the 3C9 conformation. That's why the spectrum of this cis-decalin system is found to be a narrow small lines. Actually, narrow small line is observed. Why? We know that when we consider the carbon number 1 have one axial hydrogen and one equatorial hydrogen, like this. Carbon number 2 also have axial hydrogen, equatorial hydrogen. In the similar way, carbon number 3, carbon number 4, carbon number 5, carbon number 6, carbon number 7, carbon number 8, carbon number 1, 2, 3, 4, 5, 6, 7 and 8. Each carbon having axial hydrogen, one axial hydrogen and one equatorial hydrogen. 1, 2, 3, 4 means in a ring there are 4 hydrogens in equatorial position and 4 hydrogens are in axial position. In the similar way, in B ring 4 hydrogens are in equatorial position, means total 8 hydrogens are present in equatorial position. But when we consider this hydrogen, it is equatorial. This hydrogen equatorial means total 9 hydrogens are in equatorial position and total 9 hydrogens are in axial position. But after decoupling, whatever this, suppose we consider this carbon number 1, this carbon number 1 hydrogen, which is, look at this carbon number 1, the carbon number 1 having axial hydrogen, here axial hydrogen, after ring flipping it is converted to the equatorial form, or equatorial hydrogen is now converted to the axial hydrogen. And this such type of conversion of this 9C3 conformation into the 3C9 conformation is continuously takes place at room temperature. So, because of this continuous conversion of 9C3 conformation into the 3C9 conformation in cis-decalin system, whatever equatorial hydrogens, 9 hydrogens which are in equatorial position in 9C3 conformation, after ring flipping, they are converted into the 3C9 conformation. But in 3C9 conformation, all 9 equatorial hydrogens are converted into the axial hydrogens. In the similar way, whatever 9 hydrogen which are axial position are totally converted into the 9 equatorial hydrogen. Means there is a continuous conversion of axial hydrogen into equatorial hydrogen equatorial hydrogen into the axial hydrogen. And because of this such type of continuous conversion, we cannot distinguish whether it is hydrogen is axial hydrogen or equatorial hydrogen, and therefore we observe the narrow small line in case of the cis-decalin NMR. While in case of the trans-decalin system, the two hydrogens means axial hydrogen and equatorial hydrogen clearly distinguishable, and they are coupled with each other. Therefore, the broad spectral lines, such type of broad spectral lines are observed in trans-decalin system, while such type of narrow line, very small narrow line is observed in cis-decalin system. Okay, so this will helpful to identify whether the decalin system is cis or trans-decalin system. Means NMR is one of the important tool to identify whether the system is means a decalin system is a cis-decalin system or trans-decalin